Hi, thanks for joining us today. Throughout this video, we'll be using quick sketches to outline mechanisms behind a manipulation theory utilized by the parasite T. gondii to alter human behavior. But before we get into these mechanisms, we will briefly touch on the life cycle, modes of transmission, pathophysiology, immunological responses, and other important information regarding this parasite. Let's begin with some background information on T. gondii. T. gondii is an obligate intracellular parasite that can exist in three forms: an oocyte, a tissue cyst, also known as a bradyzoite, and a tachyzoite form. It affects 1/3 of the population, though many may be asymptomatic. It does manifest, however, more in immunocompromised patients. The life cycle of T. gondii is simple, but has one specific requirement for sexual reproduction, and that is the definitive host, a feline. The life cycle begins when the feline ingests a tissue cyst, initiating infection. Once T. gondii reaches the small intestine, it penetrates the epithelial cells of the intestinal wall, begins asexual replication, and soon converts to sexual reproduction through the development of gametes. Fertilized gametes form a protective oocystic wall, and eventually the epithelial cells, which are now filled with many non-sporulated immature oocysts, burst and release the immature oocysts into the intestinal lumen to be excreted in cat feces. Once excreted, the immature oocysts remain non-infectious until sporulation occurs. This occurs faster in warmer climates and can range from a few days to several weeks. This results in the formation of two sporocysts, each with two infectious sporozoites. Transmission of T. gondii to a human host can occur one of three ways. First, and most commonly, is by ingestion of raw or undercooked meats containing tissue cysts, especially pork or lamb. Second is by ingestion of oocyst-contaminated water, milk, or unwashed vegetables and fruits. In the case of young children, accidental ingestion of oocyst-contaminated soil can occur. Lastly, pregnant mothers who contract the infection during gestation can pass it to her fetus through transplacental transmission. Once the ingested cyst or oocyst reaches the small intestine, it transforms into this fast-replicating tachyzoite form, which causes systemic dissemination of T. gondii into different tissues, where the infection becomes intracellular. Many tissues may be affected, though the protozoan has a predilection for cells of lymphoid organs, CNS, and the heart. In an otherwise healthy individual, the immune response, which we will outline next, is sufficient to reduce this parasite to the bradyzoite form, which is slow-growing and remains in the host as a chronic intracellular infection. Now we will review the immune response hosts use to control this intracellular infection. Toll-like receptors on phagocytes, such as macrophages, recognize antigen proteins. Toll-like receptor 11 recognizes profilin, and toll-like receptor 3, 7, and 9 recognize endoplasmic reticulum protein UNC93B1. Effector cells, such as neutrophils, macrophages, monocytes, and eosinophils, can generate cytotoxic molecules such as nitric oxide, granzymes, and basic proteins to kill large and small parasites. One of the most important aspects of the innate immunity, however, is the production of interleukin-12 by dendritic cells. This stimulates natural killer cells and T cells to produce interferon gamma. Together, interleukin-12 and interferon gamma induce a Th1 response, which is essential for the destruction of intracellular pathogens. When considering the adaptive immune response, CD4 positive T cells are necessary during the early stages for optimal B cell antibody production and CD8 positive T cell activation. CD8 positive cells can control infection through perforin-mediated cytotoxicity as well as production of interferon gamma. Antibodies play a critical role by opsonizing parasites for phagocytosis, blocking attachment, and also activating the complement system via the classical pathway. T. gondii, however, has specific mechanisms by which it can evade our immune system. One such mechanism is the formation of a specialized vacuole called the parasitophorus vacuole. This is a bubble-like compartment which allows the parasite to develop in the host cell while stealing nutrients and preventing lysosomal fusion. Now let's talk about treatments for toxoplasmosis, which include drugs that are inhibitors of folic acid synthesis. Most healthy people won't require treatment for T. gondii. However, if you have signs and symptoms of acute toxoplasmosis, pyrimethamine, a dihydrofolate reductase inhibitor, may be prescribed. Immunocompromised patients are prophylactically treated with trimethoprim sulfamethoxazole combo to prevent toxoplasmosis infection. Pregnant women are treated with spiramycin, which can't cross placenta, and pyrimethamine after 14 weeks of pregnancy. There is one folic acid inhibitor, however, that T. gondii has resistance to, and that is sulfadiazine. Now that we've covered basic information about this parasite, we can get into interesting new findings. While research has shown that T. gondii alters cognitive behavior and function, leading to depression, schizophrenia, and delayed response times, a fairly new phenomenon, termed the fatal attraction phenomenon, describes this parasite as a manipulator of host behavior for its own developmental benefit. 3 different pathways have been proposed surrounding the fact that once established within the host, T. gondii is able to invade immune-privileged sites such as the CNS and testes. The first mechanism of action describes tropic cysts, which rupture and recyst to evade detection. Tropism to the hypothalamus and amygdala, which are associated with decision-making and fear, could make the host more inclined to associate with Toxoplasma-infected cats. In mice, loss of fear of cat urine leads them to niches of cats and enables the transmission of T. gondii. Upon further investigation, however, it was found that even after the removal of cysts, this behavior remained, so 2 other hormonal pathways were hypothesized. The dopamine pathway is a model that suggests that T. gondii manipulates behavior by upregulating dopamine synthesis in the brain. 2 genes have been discovered in the T. gondii genome, namely AAH1 and AAH2, which resemble the enzyme tyrosine kinase, and when upregulated, catalyze the conversion of L-tyrosine to L,3,4-dihydroxyphenylalanine. Increased levels of dopamine stimulate the motivation and reward centers of the brain. This may lead to pleasurable sensation when performing behaviors that increase the transmission of T. gondii. This theory could be supported by the high correlation between toxoplasmosis infected individuals and an onset of schizophrenia. Additionally, it can be supported by findings of high levels of dopamine within cysts. Lastly, a third model refers to hormonal shifts induced by T. gondii resulting in epigenetic modification. As previously stated, T. gondii invades immunoprivileged sites such as gonads, which allow for upregulation of testosterone. Testosterone then binds to receptors on the amygdala and elicit responses to sexual urges while ignoring their defenses. Hypomethylation in the medial amygdala induces a change from activation of the fear behavior circuitry to activation of the sexual behavioral circuitry instead. This mechanism explains how T. gondii can manipulate host behavior in order to transition from the immediate host, a mouse, to its definitive host, a feline. So as we saw, T. gondii is an intracellular parasite that may elicit profound effects on human health despite previous beliefs that it simply remained in a chronic latent phase in our tissues. As we stated, there has been research to correlate this parasite with altered cognitive behavior, such as depression and schizophrenia, as well as a decrease in response time. Now we are learning that it can manipulate its hosts as well to benefit in its transmission and spread. In the end, further research is needed to find the exact mechanism by how this parasite can alter human behavior for its own benefit. While 2 theories, both hormonal, remain a possible explanation, there is not yet a consensus on what the actual mechanism is.